most people probably survive, but it doesn't matter. So please questions. So, okay, I have the first one. So I know that it was not your topic, but what's your favorite explanation for these Melanesia, Papua, and using Amazon basin, please?
My explanation is that there was probably some structure in the incoming population of the first wave, so it was not absolutely homogeneous, and that's not surprising because the land bridge was open for quite a long time. So probably there was like wave 1 and wave 1A, a later wave which for some reason survived only in the Amazon and was replaced or didn't leave any trace in other regions. And that wave is more closely related to modern East Asians and probably to modern Melanesians.
Okay. So, other question? So, okay, so, yeah, then comes up.
I just wanted to ask, I know that it's not really related to the matter, whether the Ainu people, whether there is some kind of some new information concerning their ancestry.
I know.
The Ainu people of the Hokkaido.
Honestly, I don't know much about this topic, so I cannot comment.
I thought that it might be connected to their last letter, they're quite close to that region.
Okay, I found these other areas very interesting, and I want to ask, are there any surviving populations or any sign of their approaches from today, or are they completely replaced by the neo-areas?
I mean, that's what we were talking about, whether they left at least genetic races. Concerning cultural races, it was hypothesized that a certain isolated group of Inuit in Baffin Bay, that they might be similar in terms of artifacts, in terms of cultural artifacts, to Paleo-Eskimos. But then their genetics was investigated in 2014, and it was found out that genetically they are typical Inuit, and now we demonstrate that the Nadine people have 10, 15 or 20% ancestry coming from Paleo-Eskimos. But concerning culture, again, we speculated their language might be derived from Paleo-Eskimos, and that language is distantly related to Yeniseian languages in Siberia. But that's just a speculation.
And the replacement by the neo-Inuits and also the replacement of the people by the Inuits, do you think it was really connected to the little isolation?
There is a hypothesis like that that Inuit were extremely well adapted to the north. They practiced mostly whaling and hunting seals, and they survived much better in colder environment as Paleo-Eskimos and as the north.
Then I have one, you said that Vikings lived in southern Greenland at the time when there was like replacement of one Inuit for Paleo for Nero. So do they somehow left us some information about it? Because
I don't know these issues in detail. I know that I don't think they could distinguish between types of Inuit. I guess for the Norse, all those people looked the same.
because probably the distinction would be quite notable.
It's notable for modern archaeologists and geneticists, but I doubt it was notable for the Norse.
And then I have one question. I have a question about like reducing of rare alleles. So yeah, I understand the advantage, but disadvantage is that there must be like a lot of missing drift. Like these rare alleles are lost and so on. So if you can have only like positive information there, but the negative is none. No? That if the allele is missing there, and so like how many individuals do you have to sample?
Yeah, the problem is that to obtain these positive signals you are talking about, to obtain the counts of shared alleles, you need to sample dozens and hundreds of full genomes. So, for example, you need to have a test group of at least few individuals and reference group of dozens and hundreds of individuals. So we take like one American population and compare it to Arctic or Siberian metapopulation or European metapopulation, which are much bigger. And in this way we obtain reasonable counts and that's why we obtain statistically significant differences. Of course, if we take just few genomes, we would not have any statistically significant signal. But nowadays, huge sets of full genomes are coming. Just a few weeks ago, 3 papers have been published, and in total, almost 800 high coverage genomes have been published, and now there are projects, for example, to sequence the HGDP panel, the human genome diversity panel, a widely used set of 950 individuals, so all of them would be sequenced at high coverage. So nowadays it's possible to use these methods, but we apply these methods to publish data. Obviously, we don't generate the data ourselves.
Just came to my mind, so maybe it's naive. Isn't there like higher danger of homophysing in such rare alleles? Not than frequent alleles? If it is like single SNP and so how do you deal with this?
No, actually I've not considered that possibility, but the problem is that mutations anyway are extremely rare. So usually in the time frames we consider, they don't arise many times. So they just arise once and then spread by drift. So I don't think that homoplasia is a big problem for this type of data.
So, any other question? Yes, now it's time. If not, so thank you very much once more.
Thank you.